Hello, everyone. So today we're going to be talking about antibiotic therapy, so antimicrobials. So just so we understand, so antibiotics generally are going to kill, there is a few that will kill other things, but generally bacteria. Some of them will also kill some protozoa. And the way to do antibiotic therapy, you need to generally there is assumptions that are made of what kind of bacteria we're talking about, but the best way to do antibiotic therapy is to actually know what we're dealing with, so you can pick antibiotics that will specifically kill what you're trying to kill. So let me do this guy here. Okay. So we're going to talk about 4 different classes of antibiotics, but antibiotics are divided generally into, there's 4 classes, but there's just 2 ways that antibiotics can be divided. And one big list is what we call bacteriostatic, which bacteriostatic will only prevent the growth of more bacteria, and bactericidal will actually kill the existent bacteria also, okay? So this graph here is showing what each antibiotic type will do. So this, is time, so how many bacteria are growing. This is the number of viable bacteria. As you can see, without any drugs, this is how they grow. It gets to a point, you see that they plateau. The reason why they plateau is because they ran out of space or food for that dish, okay, for that little plate. And here is when they added different drugs. When you add a bactericidal agent, you see the log of viable bacteria. I don't know what this thing, and I'm trying, by the way, to figure out why this does this, okay? When you add a bactericidal agent, you see the log number of bacteria decrease, go back to 0. And when we add a bacteriostatic agent, they just stop growing, okay? So the bacteriostatic agent generally you take for longer periods of time, or you have to know exactly what you're dealing with, or generally you take for longer and allow the body to also start fighting the bacteria themselves, okay? There is other things. So antibiotic therapy, I don't know if you guys know this, but it's ideally the antibiotics are taken at the same time every day. So for example, if an antibiotic is supposed to be taken once a day, so let's say 8 in the morning, it's supposed to be taken at 8 in the morning every day. It can be 8 today and then noon tomorrow and then 6 p.m. the day after and then 8 again, because antibiotics, they work in different ways. Let me share here the screen. Where is the whiteboard? So antibiotics are going to work in different ways, and we're going to have one that is time dependent and different types. Some of them are time dependent and some of them are dose dependent, okay? And the ones that are time dependent, for example, amoxicillin that you have to take, or cephalosporin that you have to take 3-4 times a day, that means that here's the level in the blood of antibiotic that kills the bacteria. You have to take 4 times a day because it goes like this, 4 times a day, and it reaches the blood. So you want more or less that each time that you take the drug to stay, it's only killing bacteria and it's above this level, okay? So that's why it's important that you don't allow this particular antibiotic. Let me go a different color here, hold on. To go, if you take today, say at 8 a.m., and then tomorrow at noon, it's going to be like this. It's going to go too low, and then you take it again. It's going to rise here, and then you take like too quickly. It's going to come here. So if you take like at noon, and then the next day you take like at 6 a.m., it's going to come here, and then you wait too long. It does this. So this doesn't work. Okay, you have to take antibiotics. Eraser. You have to take antibiotics at the correct time. So this is the time-dependent antibiotic, okay? The dose-dependent antibiotic works a little bit different. It's the ones that you're going to take maybe once or twice a day, generally once a day. So it's like Levaquin or enrofloxacin, etc. And that antibiotic means this is still the level that kills the bacteria. But this one is gonna, once you take it, reaches super, super high and kills a ton of bacteria, and they're still knocked out. And then you take it again the next day and it goes really high again. And those are what we call dose dependent. Okay, so the dose dependent antibiotic you just give and it reaches the blood circulation very, very quickly and in a dose that is above very quickly, and it kills a ton of bacteria during this time that is above the line. And it's enough to take it once a day, because even if it goes under the line and it's not killing bacteria right at the moment, once you go up again, it's going to stay above the line for a long period of time. Okay, so that's why it works like that. So it's important anytime that we're doing antibiotic therapy to do exactly like this. Okay, to do at, I feel that doctors don't tell us that, and they just say take this 3 times a day, and people may take like at 9, noon, and 3. That doesn't work. If it's 3 times a day, 24 hours divided by 3 is 8 hours. So it's every 8 hours. Okay, guys. So that's important for you guys to understand. Now, bacteria, there's multiple different ways to classify bacteria, but one of the main divisions in bacteria is if they're gram-positive or gram-negative. And that has to do with their cell wall. Okay, so the gram-positive bacteria have a different type of cell wall, and the gram-negative have a different type, and they come with, each one come with their problems. Okay, so the gram stain is you put the bacteria on a little plate, and then you do the stain. I guess I'm hoping you guys learned this in biology, but you stain with crystal violet, which is purple. Then you wash with iodine. For 30 seconds. Iodine is going to be a little like pinkish color, and then you are going to wash with acetone to remove all, and then you're going to remove all stain that hasn't stained, and then you're going to put safranine, and that's going to stain the gram-negative bacteria. So this is how it turns out. So it turns out that the ones that are gram-positive are going to stain in blue or purple, dark purple, and the gram-negative are going to stain in this rust color. It's a red kind of rust color, and that's because of the safranine. Do I need to know the different bacteria? Not necessarily, but I want you guys to understand the problems of gram-positive, gram-negative, and how at least the most important bacteria, what they are. So, for example, staph and strep, they are gram-positive. Okay, they should be easy to kill. Okay, E. coli is gram-negative. I want you guys to understand that Clostridium is gram-positive. These particular ones here on this area are aerobic, and these ones here are anaerobic. So anaerobic means that they live in the absence of oxygen, and aerobic means that they live in the presence of oxygen. And then there is also facultative anaerobic, which means that they can live either way. Okay, either in the presence or absence of oxygen. Pseudomonas, very common to have. This thing lives everywhere. Anyway, I don't see salmonella here, but salmonella is gram-negative. Okay, so this is a schematic of the cell walls of the gram-positive and the gram-negative bacteria. So the gram-positive they're going to have a bigger cell wall. So we're going to see here that this is the cell membrane, and this is one of the things. So mammalian tissue and animal tissue, not just mammalian, but like chickens, also fish, they have cell membranes. Okay, so animals have cell membrane, which is the lipoprotein membrane, the cytoplasmic membrane. Bacteria have that, and some bacteria, all bacteria, they have either a cell wall that is a gram-positive, or they also have a cell wall that's gram-negative. So they have this extra layer around them that's called a cell wall. We don't have that extra layer. So what's the importance of that? We're going to see that one of the important points in antibiotic therapy is to harm the pathogen, but not harm the host. So how we pick antibiotics, so hopefully antibiotics are going to have higher affinity for something that happens in the pathogen, but not something that happens in the host. So an antibiotic that attacks the cell wall, for sure, is not going to do anything to the host cells because we don't have a cell wall, okay? So you're going to see here, this is the cell wall for gram-positive bacteria. This is the gram-negative cell wall here. And gram-negative, they have this thing here called lipopolysaccharide. And lipopolysaccharide is going to be something that is going to come back to us because this is what causes a lot of the toxicity in gram-negative bacteria. Is a lipopolysaccharide can actually, every time gram-negative bacteria, they die or divide, they release endotoxins, and LPS, lipopolysaccharide, is one of the most important ones, okay? So this is, so just to give you, am I gonna ask specific questions about their cell walls? No, but you need to understand that gram-negative are different from gram-positive, and LPS is a problem, and antibiotics that work more towards gram-positive or towards gram-negative. So we're going to have 4 different classes of antibiotics. So number 1 is the oldest type, so cell wall synthesis inhibitors. They're going to be the beta-lactams, which is the penicillins, and cephalosporins, which are very, very similar to penicillins. And there's going to be other drugs also that are cell wall synthesis inhibitors. We're going to have protein synthesis inhibitors. And those are the ones that are going to work on the ribosomes. And if you guys remember, mammalian or eukaryotes, ribosomes are different. We have the 80-40, yeah, and bacteria are going to have the 70-30 ribosomes, and so they are going to have affinity more for the bacterial ribosomes. But because it's ribosomes and we also have ribosomes, every once in a while it can actually affect us as well, okay? Or mammalians. Macrolides also are protein synthesis inhibitors, and then we're going to have inhibitors of the metabolism. They are going to inhibit the formation of folic acid, which is food for bacteria, and those are going to be your sulfa and trimethoprim. And then the latest type of antibiotics is the DNA synthesis inhibitors, and they're called also the quinolones. This is the latest, most modern type of antibiotic, but also the one that it's so powerful that we prefer to not use it, except in cases that the animal is septic and is not responding to any other antibiotics. So this is just a schematic of a bacteria. So you can see how it has a plasma membrane, it has a cell wall, ribosomes. Bacteria don't have a nucleus, so they just have DNA material or nuclear material loose in the cytoplasm, as opposed to us, okay? And this, again, is just a schematic of the bacteria and where each one of the antibiotics are going to work on. And so inhibitors of metabolism, that's the folic acid, cell wall synthesis inhibitor, something that we don't have. Protein synthesis, so that's the ones that are going to work on the ribosomes, and then the inhibitors of nucleic acid function and synthesis. Principles of antibiotic therapy: ideally you want to harm the parasite, so the pathogen, and not the host, okay? So we need, when treating with antibiotics, we want the least side effects possible, okay? So the way to do this, hopefully use antibiotics that are going to have a process that is unique to the bacteria and not to us, okay? So that's when cell wall synthesis inhibitors, because we don't have cell wall, it's a very safe antibiotic, except it can actually cause allergy. So like people that are allergic to penicillin can actually die, but it's not toxic. It is sensitizing, meaning it can create allergy in animals, but it's not toxic. So it doesn't like, there's going to be antibiotics that are toxic to the individual, such as like ototoxic, it kills the hearing, nephrotoxic, it kills the kidneys, etc. Processes that are common to bacteria and host cells are trying to develop antibiotics that have more affinity for bacteria. So that's when I talked about the ribosome. So hopefully we'll attack more the bacterial ribosomes as opposed to ours. And then you need to figure out what type of bacteria we are talking about, and that's when the antibiotic is going to be chosen like that. So cell wall synthesis inhibitors, we're going to start with the beta-lactam antibiotics, which are the penicillins. Penicillins were discovered 100 years ago, okay? Very old antibiotic, the oldest one. It is very safe to be used most of the times. It is one of the things that it's a problem is that it can be sensitizing, okay? So meaning it can develop into allergies. And once you're allergic to penicillin, you are not allergic to it, okay? Important is that it's unstable. I don't know why it does this. Do you guys know? It is unstable in acidic pH, which just means that it's important because like pus is very acidic. So penicillin doesn't really work well where there is pus. So if there's a lot of pus, you have to remove all that, even if it's gram-positive bacteria, and then the penicillin can be effective then. However, the other thing too, because it is not very stable in acidic pH, just the oldest type of penicillin, you're going to see, you cannot take it orally because the stomach is very acidic. And then as they created more synthetic type of penicillin, such as amoxicillin, ampicillin, those are more stable in acidic pH and can be taken orally, okay? But penicillin in general cannot. The other thing too, they are beta-lactam antibiotics, which we're gonna see here in a second. The beta-lactam is this ring here. They have this ring, as you can see. So amoxicillin has this ring, ampicillin has this ring. So all beta-lactam antibiotics, why the heck does it do that? All antibiotics, beta-lactam antibiotics are gonna have this, and then they're just gonna change here in the radical, okay? So where we see here, so as you can see, NH2 or NH2, NH here, NH here, and this radical here is different from this guy here. Like this one has an extra hydroxy group. This one doesn't have an extra hydroxy group. So this is what this R represents, is the radical, okay? The interesting thing about penicillins and beta-lactam antibiotics is that some bacteria have this enzyme called a beta-lactamase, and they can actually destroy the penicillins. So beta-lactamase is this enzyme that goes and breaks that beta-lactam ring. And when that happens, it can actually cause what we call, I know it's a funny name, superinfection. Superinfection is that almost like these bacteria are going to feed off of this antibiotic. They're going to multiply like crazy, and they can actually cause very severe diarrhea in these horses. So this is one of the things because of the beta-lactam, and some bacteria have the beta-lactamase, not all. There's different formulations for penicillin. It is going to not allow the bacteria to form the cell wall. So that way they cannot actually, as they start to try to multiply, they can't because there is no cell wall being made. So they die off, okay? And the bacteria, some of the most important bacteria that have beta-lactamase, Staph aureus, okay? As it's like very common to have surface infections with those. E. coli, which is a GI tract kind of bacteria, Escherichia coli. And E. coli is actually gram-negative, so penicillin doesn't even kill it, but in the presence of penicillin, it just goes and starts to kill the penicillin and starts to multiply really quickly, forming what we call superinfection, okay? Penicillin is the drug of choice to treat infections caused by Streptococcus, okay? Meningococcus, what is meningococcus? Causing the bacteria that causes meningitis. And like Clostridium is a gram-positive bacteria, Anthrax, gram-positive. Side effect is not toxic, okay? We're gonna see some other antibiotics here that are toxic. The serious side effects is generally because of hypersensitivity, which is the same as allergy, okay? Causing anaphylactic shock, which can lead to death, okay? Other side effect is the superinfection when they encounter the beta-lactamase producing bacteria. Resistance, because it's one of the oldest antibiotics, so nowadays there's a lot of resistance to penicillin, especially in human medicine, but it's very, very much used in horses. I guess in humans, people don't totally use penicillins anymore in case it's a person that is sensitive and they can actually die of anaphylactic shock. But like amoxicillin is very much used, and it's a synthetic penicillin, very safe, okay? Semi-synthetic penicillin, this is what I said here: ampicillin, amoxicillin, methicillin. You guys have all heard of MRSA, which is methicillin-resistant Staphylococcus aureus, which is a type of Staphylococcus aureus that is resistant to a lot of antibiotics and can cause very, very, very serious skin infections and other infections as well that can kill a person, okay? Already talked about that. Other types of beta-lactams would be cephalosporins, very similar to penicillin in the mechanism of action and toxicity. It is a little bit more stable than penicillin in the bacterial beta-lactamase, which makes the cephalosporin a little bit broader spectrum, okay? Penicillin is more narrow, only for gram-positive, okay? Generally for gram-positive bacteria. So because this one is a little bit more stable, it has a broader spectrum. Now, the other thing too is that cephalosporins are divided into first, second, third, and fourth generation. And the first generation is more active towards gram-positive bacteria. And then as they go towards the fourth generation, it becomes active against gram-negative bacteria, okay? So it makes it a very a nice group of antibiotics to use because it can work towards gram-positive and gram-negative according to the type of antibiotic. The thing about cephalosporins in horses is that it is an expensive antibiotic. So it's not used all the time, but it is an expensive antibiotic. And when useful, it is actually very useful. The other thing too is that the beautiful thing is that it crosses the blood-brain barrier. What is blood-brain barrier? It is this: very few things can cross the blood-brain barrier. It's a barrier that exists between the blood and the brain, which is not everything that is in the blood that actually can get to the brain, okay? So, for example, medications that we take, or like recreational drugs that we take that are hallucinogenic, obviously you take it, it goes to the blood and it goes to your brain. So that crosses the blood-brain barrier, but other medications don't, okay? Or other drugs actually don't either. So the importance of crossing the blood-brain barrier is because of its use when you have infection of the brain. So something that is necessary if you have meningitis or any other kind of infection of the brain, it's important to have a medication that is able to cross. It can be used when you have penicillin sensitivity. You could actually use cephalosporins instead because especially if it has the same effect or is trying to kill the same type of bacteria, but they can also be sensitizing, and you can give this, orally, IM, and IV. And here is another, again, oh my gosh, I need to figure out why this does, the beta-lactam ring. So other antibiotics, they can be other beta-lactam, or they can also, sometimes we add beta-lactamase inhibitors, okay? So that is to inhibit that enzyme from the bacteria. And one of them is this guy right here, clavulanic acid. So that, if you guys have taken the medication called Augmentin, that's amoxicillin plus clavulanic acid, because when you add clavulanic acid to the mix, you're adding something to prevent the bacteria from breaking it up. And so that means it has better action. Just amoxicillin, maybe it's very simple. There's bacteria that are going to be resistant to it. When you add the clavulanic acid, it increases its more broad spectrum, okay? Vancomycin is a very important type of antibiotic, but it's generally only used in hospital setting because it can be toxic, okay? And then another one that is important, and I need you guys to know this by heart, bacitracin. Bacitracin is a beta-lactam antibiotic, but it's very, very toxic also. And because it's toxic, it is generally combined with polymyxin B and neomycin in what we call triple antibiotic ointment, okay? So these three antibiotics, bacitracin, polymyxin, and neomycin, are wonderful. They kill bacteria really well, but they can be toxic. And therefore, it's better they were created to make into the triple antibiotic ointment, which can kill surface bacteria very well. It's not anything that you put topically generally is not very much absorbed, and that's how you can curtail the toxicity. So it's important that you guys memorize bacitracin, polymyxin, and polymyxin B and neomycin, because that is what makes the triple antibiotic ointment, okay? What else did I want to say about this? The other thing that's important also about antibiotic therapy is that you need to finish the course of antibiotics. And the way that they work is you have sensitive, what the heck? Sensitive bacteria and a few resistant bacteria in any disease. The first round of antibiotics, you kill a lot of the sensitive, and then as you increase more time and more dose, higher dose or longer treatment, that's when you kill the resistant bacteria. If you finish antibiotic therapy without killing those two, and these two maintain alive, then you have these resistant bacteria. These two are going to start to multiply, and now they're resistant to that antibiotic, and now that antibiotic is not going to work for that infection anymore. And once you have the resistant type of bacteria, very difficult to come up. They don't become unresistant, okay? Protein synthesis inhibitors, we're talking about the tetracyclines and the macrolides. Tetracyclines, they are broad spectrum. They're wonderful medication. They will work with gram-positive, gram-negative, some rickettsia, anaerobes, protozoa, etc. So tetracycline 1 that we use very much is what we call LA200, which is oxytetracycline. We also use doxycycline, and we also use, but doxycycline is used in horses as well. In humans, we use also something called minocycline, so that's important. It accumulates inside the bacterial cell and is going to bind to the ribosomes, impeding the formation of new protein, okay? And that's how tetracyclines are generally bacteriostatic. They don't kill the bacteria, they just prevent the bacteria from forming protein, and then they die off that way, okay? But they don't necessarily kill the bacteria right then. The important thing about tetracyclines is that it chelates calcium and other divalent cations, okay? So chelates, it means that it joins, it binds up to calcium, and that can be a problem because, here's why. Number one, if you're taking this, you should never take with milk, because if you take it with milk, the tetracycline will bind to the milk, and then you depend on that concentration in the blood, but because it already bound to the milk, to the calcium in the milk, you're going to have less available in your blood. Another divalent cation is magnesium. Okay, divalent means that it has the 2+. So like calcium has a 2+, magnesium also has the 2+ as well, okay? The other thing about chelating calcium is that you should never give this or take this if you are a growing individual, okay? So foals cannot receive this because they are growing and they need a lot of calcium being deposited in their bones. So if it chelates calcium, it can become a problem, okay? The other thing too about this is when giving this particular antibiotic via IV, generally that's how you give to horses. You give IV for the oxytetracycline. For doxycycline, it's oral. But when giving IV, you have to give a very, very slow injection so you don't have a bolus of this drug that chelates calcium, and when it passes through the heart as a bolus, it can actually stop the heart, and that can become a problem. So you give very slowly. Some veterinarians get the dose, and then they also add more saline to the dose to be able to give very slowly, or just give it very, very slowly so it doesn't, as it gets to the heart, it doesn't stop the heart, okay? Like I said, should be avoided in young or pregnant individuals. So the reason why it's called tetracycline is because it has these 4 rings. Tetra is obviously means 4, okay? Macrolides are also protein synthesis inhibitors, and this is probably a type of antibiotic that's very common to us. If you have had like sinus infections and you have taken the Z-Pak, so Z-Pak is this guy right here, azithromycin, okay? So macrolides are going to be erythromycin, clarithromycin, azithromycin, etc., okay? They are very useful in human medicine, not so much used in horses because it can cause very severe diarrhea in older horses. So if you're older than 1 year old, it's not used in horses. Because it can cause this very severe diarrhea. The other thing that it can also cause is light sensitivity. So it's photosensitizing. So once you're taking this, and this goes for us also, you should not be going out in the sun because you can actually burn much more than you would normally. And in foals, if they're going to be taking this, because it is one of the drugs of choices to treat this pneumonia in foals caused by Rhodococcus equi. But these foals, if they're taking erythromycin, should avoid being exposed to the sun, especially if they have a lot of white in them, because they will burn, okay? Now, erythromycin is used to treat Rhodococcus in conjunction with rifampin, and there is other drugs also that can be used to treat Rhodococcus, but it's just lucky that this one, erythromycin, is useful, and this is a disease that only affects foals because this particular drug cannot be used in adult animals. Other protein synthesis inhibitors are going to include aminoglycosides, and they're going to be streptomycin, neomycin, gentamicin, amikacin, tropramycin, okay? So if you guys remember, neomycin is part of Neopolybac, okay? So it's part of the triple antibiotic ointment. Gentamicin is very good against gram-negative bacteria, and it can generally be used in conjunction with another type of antibiotic, say sulfotrimethoprim, to try to be more broad. Sulfotrimethoprim is going to kill more gram-positive, gentamicin gram-negative. Amikacin is a wonderful antibiotic too, but it can be very toxic. So all these guys here, all these aminoglycosides can be toxic, okay? They are toxic. So they are used very only in like hospital settings or only used with veterinarian assistance. Amikacin, they sometimes they get included in joint injections. So if you're gonna inject the joints of the horses because of arthritis, when you're gonna do the steroid plus hyaluronic acid, sometimes you include a little bit of amikacin to prevent the joint from blowing up. You can also add amikacin to do joint lavages, so that works also. So they are very, very, they kill a lot of bacteria. However, they are, this is important, the reason why we don't use them a lot is because they are extremely ototoxic, and nephrotoxic. So they will destroy the hearing and they will destroy the kidneys. Okay, so that's why they are not used on a day-to-day medicine. So amikacin is used in horses. Obviously, neomycin is used in horses in the triple antibiotic ointment. Gentamicin is used in horses, but it's not something that you just buy at the pharmacy. It's definitely not like SMZ TMP tablets, because they are more problematic. The other class is the folic acid synthesis inhibitors, and those are going to be the sulfa and trimethoprim. Sulfa is going to be structurally similar to PABA, and PABA is what these microorganisms are going to use to form folic acid, which is essential for nucleic acid synthesis. So they grab PABA, bring to the inside of themselves, and they use that to form folic acid. Sulfas are going to be structurally similar to PABA. So in the presence of sulfa, this bacteria, instead of grabbing PABA, may grab sulfa, but guess what? Once they bring sulfa inside, it's not PABA, so they cannot form folic acid because it was sulfa. Sulfa in itself is bacteriostatic, so it just doesn't allow the bacteria it doesn't kill the bacteria, it doesn't allow the bacteria to continue to multiply and create bigger infections. And that's why, to make this antibiotic, generally antibiotics are worked in one. You do generally one antibiotic instead of multiple because of different mechanisms of action. And, so you don't want to cause resistance. But in this particular case, sulfa is actually more useful if added to trimethoprim. And then they together will become bactericidal. And that's why I said here's not used by itself anymore, but with trimethoprim is used every day in our lives, okay? And that's when sulfa and trimethoprim become bactericidal, because they have this synergistic effect with each other. And then the last type of, or the last class of antibiotics is going to be the DNA synthesis inhibitors, or the quinolones. And they are the latest antibiotics, but they're not that new either. They're like 25, 30 years old. Maybe 40, I don't know. And they are the floxacins. So if, ciprofloxacin, enrofloxacin, they are going to be the ones. And they are fluoroquinolones, and they are very broad spectrum. They will do a lot of harm to bacteria. So that's why we only use them. They are like the machine gun of antibiotics. And generally, you want to use one that's more narrow to kill exactly the bacteria that's causing the problem. And that's why doing a culture and antibiogram is important. But when you don't know and the animal is septic and is going to die anyway, you do something that is more broad spectrum. It is newer. It is also very useful towards gram-positive and gram-negative. It's what's used to treat MRSA these days. And the one thing about the floxacins is that it can actually cause a problem with tendons, okay? And it can actually destroy, so it's not toxic to the ears or kidneys, but it can destroy tendons. So generally, we don't use floxacins in young growing animals, so it doesn't destroy their tendons, and it's generally not for long periods of time either. Now, what's interesting is that the medication that we have in the United States that is a floxacin is only approved for cattle, and it's called Baytril. So veterinarians will use Baytril off-label to treat some infections in horses when it's not responding to anything else, especially gram-negative infections. So Baytril is a cattle medication, and it's used off-label with veterinarians in the United States. Now, in other countries, they have Baytril is also allowed in horses as well. So this is antibiotic therapy. If you have any questions, don't hesitate to contact me.